Hi everyone, welcome back. In a world heavy with antibiotic use, resistance is key. Species and strains require resistance in order to survive exposure to antibiotics. Organisms have to deal with the conditions of their environment, and resistance to those conditions is key to survival. When antibiotics are part of that environment, resistance to antibiotics is also a key trait that species have to be successful in that environment. Thus, it should come as no surprise that as penicillin rose in prominence and use, penicillin resistance followed very quickly. The antibiotic was first available in pharmacies in 1945, but the first reports of that resistance were in 1947. Mutations emerged and the species evolved when those mutations were suddenly useful. This is not just some trend that we have seen. This is the expectation. When the population varies in its vulnerability or resistance to some quality of its environment, the individuals that can survive better, that are more resistant to those conditions, are going to do better. They're going to survive longer and reproduce more than those that are more vulnerable. Now, before we really talk about antibiotic resistance in nature, I want to discuss how antibiotics work on a microbial species. Because antibiotics have different kinds of modes of action. Essentially, how the antibiotic is going to impact the cell, what specifically it attacks or interferes with in order to result in its death. Different drugs are going to hit different anatomical features or physiological characteristics of a cell. For example, penicillin attacks the cell wall. So if you think back to our early discussions about the configurations of bacterial cell walls, you may remember that peptidoglycan is made up of chains of alternating sugars that then form these crosslinks with each other, and those crosslinks are made of proteins. Now, penicillin prevents those crosslinks from forming, considerably weakening the cell wall and allowing for it to develop massive holes as the cell wall is being repaired. Now, the function of penicillin actually comes from its molecular structure. The compound has a certain four-membered ring in the center of the molecule called a beta-lactam ring. We call it four-membered because it's made of four atoms, right? We can see it right here. Now, this ring fits really well in the active site of a specific enzyme that bacteria have called DD transpeptidase. This is the enzyme that builds those protein crosslinks in peptidoglycan. So penicillin competitively binds to that active site of DD transpeptidase. The bacterial cell wall constantly has to be remodeled. As the bacteria grows and divides, as it takes any injuries, DD transpeptidase is constantly working so that the bacteria doesn't have any permanent lasting holes in the cell wall. That can create a lot of problems and compromise the integrity of the cell. When we introduce penicillin, we get that competitive binding, and DD transpeptidase is prevented from doing that job. As a result, these holes develop, and that can cause the cell to rupture. Now, penicillin is what we would call a natural product, something that is made by another organism. In this case, this is a fungal product made by fungal enzymes. Now, by preventing peptidoglycan from forming these crosslinks, penicillin can actually kill these bacteria. It does not solely inhibit their growth, but it actually leads directly to their death. This is what we would call a bactericidal drug. So penicillin is a great example of this. A drug that directly leads to bacterial death. It has some kind of killing ability. This stands opposed to something we would call a bacteriostatic drug, a drug that inhibits the growth or function of bacteria but does not kill them. So bacteriostatic drugs, through their inhibitory function, they stop the bacteria from doing their normal life cycle routines and reproducing, but the cells still remain. They still exist until they eventually die of other means. This is an important terminology, the difference between something that is cidal or static. We see the same terminology used in other kinds of antimicrobials, like fungicide versus a fungistatic, a virucidal versus a virostatic, and so on. And we can see the impact of this really well in a petri dish. Of course, a petri dish is a more exaggerated condition than what we see in real life. The human body is not just a literal petri dish, but we can see what this looks like in a perfect system. Here we have antibiotic that we administer to the site, and it permeates through this gel. And so bacteria that grow on that gel are being exposed to that antibiotic, and there is no growth. Any bacteria that establish there die out. This is what we call a zone of inhibition as it forms. It is a dead space that exists, a no man's land, between where the bacteria can grow and where it cannot grow. On a petri dish, the antibiotic, when we apply it, it spreads out from the point of application. It spreads through the gel, diffusing through it, moving as always with diffusion from a point of high concentration at the site of application to a point of low concentration further and further away from that point of application. At a certain distance, as this antibiotic diffuses through the environment, the growth will not be inhibited. Essentially, when the antibiotic is low enough in concentration that it's not deadly for the bacteria anymore. This is what we call the minimum effective concentration, the concentration in which the antibiotic starts to be effective. This is something we can actually test in the laboratory, of course, the inhibitory effect of a specific drug. We use something called a Kirby-Bauer test. So to do this, we have a medium, and we have these little paper discs that we can soak with antibiotic. And then we can apply them to the plate and allow the diffusion of that antibiotic through the media. As it moves out from that point, we're going to potentially see zones of inhibition form depending on how deadly this antibiotic is for the bacteria that we applied it to. And that zone of inhibition can be measured. The size of that zone of inhibition is directly related to the drug's concentration. As we said, the closer you are to the application source, to that little disc, the higher the concentration is. The further out from that disc, the lower the concentration is. We can use this to figure out the minimum effective concentration for this drug for a species. And the starting concentration of the antibiotic in that disc also matters. If you start out with a very low concentration, the zone of inhibition is likely going to be smaller. If you start out with a higher concentration in that disc, there is simply more antibiotic to diffuse through the media, and so that zone of inhibition can be larger. Now, antibiotics can also be modified as well to do extra jobs or have a modified function. A lot of these antibiotics have accessory parts or side chains that can be altered in a lab, and that can allow us to change the structure of these chemicals, which in turn changes their function. We can now create what we call semi-synthetic drugs, these derivative compounds that we produce using an original natural product. Like penicillin, for example, we can change the side chain on it, add things to it to modify the function to make it more effective, to make it able to get around an antibiotic-resistant strain's defenses, or have it target a wider spectrum of bacteria, and more. But also we can make fully synthetic drugs as well. We could create some drugs fully in the lab using not a single living thing or their enzymes to produce it. We could do so just with chemical reactions in a laboratory setting. There are some drugs that we construct this way. Now, a good antibiotic has to have a few key qualities. It should be selectively toxic, meaning the antibiotic should attack really just the pathogen and not harm our cells. It needs to target the things that we want to and not cause harm to the body. It should also be complementary to host defenses. It should be something that helps us defend ourselves and not get in the way of our own immune system. It needs to work synergistically with us, not antagonistically. It also needs to remain active in the host's body. So when we are treated with that drug, it has to remain functional until that infection is gone. We should also be able to take it and distribute it where it needs to go. So if you have a topical infection, you should be able to apply that drug topically. If you have a widespread infection, you need to be able to administer this antibiotic widely throughout the body. It also really needs to be shelf stable, ideally. Something that we could store and hold for a long period of time, including before it gets prescribed. It has to be accessible this way. And most importantly, well, it has to work. It has to actually do the job we want it to do, fighting the thing we need it to fight. On these modes of action, there are many different ways that antibiotics can work. Drugs like penicillin and bacitracin and vancomycin attack the cell wall. You can attack the synthesis of the cell wall or the repair of the cell wall. You can attack a variety of different features of that structure. There are other drugs like tetracyclines and macrolides that will inhibit the ability for bacteria to produce their own proteins. In this way, they are disabling the major functions of that cell, and that can also lead to the cell eventually dying. It can break the cell's ability to create new DNA or RNA. The RNA, of course, is critical for them to be able to make more proteins, but the DNA is critical for them to reproduce. Now, likewise, these antibacterials can differ in whether they outright destroy or inhibit the cell's function. So some drugs will do one, some drugs will do the other. Things that inhibit the cell's reproduction are what we would call bactericidal. These are more inhibitory. Whereas things that might attack like the cell's key structures or main functions and prevent it from doing homeostasis and surviving, those things usually are bactericidal. They will outright kill the bacteria. We could apply the same examination to antivirals and other kinds of drugs. We have similarly virucidal and virustatic drugs that can attack the virus, either preventing it from reproducing or destroy the particles. So there are different methods we could use. For example, if the virus has an envelope around it, we could attack that. We could attack the virus's DNA or RNA. We could attack the virus's main mechanisms of assembly and escape from the cell after it has reproduced. Similarly, fungicidal and fungistatic drugs, we can attack their nucleic acid synthesis, we could attack the cell membrane, we could attack their cell wall as well. We could also attack their ability to reproduce. Antibiotics ultimately fall under a spectrum of action. They could be either broad spectrum or narrow spectrum, where broad spectrum means they are quite general, very non-specific, and will attack kind of a wide class of bacteria. Narrow spectrum antibiotics are much more specific, usually targeting only a handful of species that share a very specific characteristic. Now, we generally, if we can, prefer to use narrow spectrum antibiotics over broad spectrum antibiotics when possible. Narrow spectrum antibiotics are naturally very specific, and so if we can use those specific antibiotics first before relying on the broader antibiotics, we can hopefully avoid risks that are associated with those broad spectrum antibiotics. Namely, if you apply a broad spectrum antibiotic, you do have a wider range of bacteria that you attack, including friendly bacteria that can be present in an organism. That can change the person's microbiome. But also, we really want to avoid driving antibiotic resistance when possible. Broad spectrum antibiotics expose such a wide collection of bacteria to the antibiotic that can be a driver of antibiotic resistance, not just in the species that we're trying to get rid of, but also in all of these other species that live in or on the patient. But this also carries with it some challenges, because if we're going to use narrow spectrum antibiotics, we have to know the pathogen that is causing the disease. We need to know it down to species. And that isn't always possible to do really quickly. There are some diseases that produce symptoms that are similar to a wide variety of other diseases. And identifying the causative agent of that disease takes time, money, energy, and resources. Those things are not always available in one form or another. So a lot of time, we often have to fall back on these broad spectrum antibiotics, even though we would prefer not to.